Hello everyone. So the next topic which I will be discussing is about the transmission of impulse. Now the transmission of impulse involves three steps, that is polarization, depolarization and repolarization. So we will be discussing each of these steps in detail. So let's start with the introduction first. That the transmission of the nerve impulse along a neuron from one end to the other occurs as a result of electrical changes across the membrane of the neuron. So what is it? It is the result of electrical changes that takes place across the membrane of the neuron. Now, there are three steps, as I have already told you that there are three steps that are involved in the transmitting of the nerve impulse. First is the polarization, next is the depolarization, and the third is the repolarization. Now, here is a diagram which is representing all the three steps in detail. So first step depicting is here about the polarization, which is also known as the resting potential. The second stage is known as the depolarization, which is known as the action potential. And the last is known as the repolarization. So we will be discussing each of these stages and will able to understand this diagram in a proper manner. Now, first stage is the resting potential, which is known as the polarization stage. So in this, a neuron at the resting is electrically charged, but it is not conducting. So a neuron in this phase is electrically charged, but it is not conducting. So there is a plasma membrane or exoplasm, which is the part of the neuron, and it is negatively charged as compared to the intestinal fluid. Now the potential difference which is measured at this stage is called as the resting potential, which is about minus 70 millivolts. So the intestinal fluid, they have a high concentration of Na plus, like sodium ions, which was being depicted in the figure as well, which is about 16 times higher outside the neuron than inside the neuron. So it is 16 times higher than that of the outside of the neuron than the inside of the neuron. So similarly, the exoplasm or the plasma membrane which we depicted, they have a high concentration of potassium ions, which is about 25 times higher inside than outside the intestinal fluid. Now, due to this difference in concentration of the ions, the Na ions, the sodium ions, they tend to diffuse into the exoplasm, and the potassium ion tend to diffuse outside the exoplasm. So the membrane of neuron at the resting is more permeable to potassium ions than the sodium ions. So the potassium ions leaves the neuron faster as compared to the sodium ions which enters the neuron. So the difference in the permeability results in the accumulation of high concentration of cation. What are cations? Cations, they are the positively charged ions outside the neuron as compared to the concentration of the cation that is inside. So this stage of resting neuron is called the polarized state, and it is electronegatively charged. So this stage is electronegatively charged. So the next phase which we will be discussing is about the depolarization state, which is also called as the action potential. So any stimulus which is beyond the threshold can initiate an impulse. So when such impulse is applied in the resting neuron, it opens the sodium channel. So now the permeability of the sodium ion will suddenly increase at the point of the stimulus, which is causing the depolarization. So what is happening? When the permeability of the sodium ion, it will increase. So when there is an increase in the permeability of the sodium ions, at the point of stimulus, it will cause the depolarization, which we are discussing here. So what is it? So the diffusion of the sodium ions increases by 10 times from outside to inside. So as a result, this plasma layer becomes positively charged, which is exact opposite to polarized state. So it is called as the depolarized state or reverse polarized state. So this depolarization of membrane stimulates the adjacent voltage channel, so the action potential passes as a wave along the length of the neuron. So this was about the depolarization. Next is about the repolarization. So when the concentration of the sodium ions inside the exoplasm increases, there is a permeability to sodium ions which decreases, and there is a sodium channel which starts to close. The sodium channel here will starts to close, but the sodium and potassium pump activates so that Na plus are pumped out and K plus, that is the potassium plus, inside until the resting potential is being restored. So this process is known as the repolarization, as it starts from the same point where the depolarization starts. So it will start from the same point. So the entire process of polarization, depolarization and repolarization occurs within fraction of seconds. So it will occur in fraction of seconds. Now again the neuron is read for another impulse. So these three processes will happen in seconds. The next is about the synapse. So here is a diagrammatic representation which is showing the synapse, and different parts of the synapse are being labeled here. So synapse also helps in the transmission of the neuron impulse, nerve impulse. So the place where an axon terminal meets the another cell, that term is known as the synapse. So each of the terms which was being depicted in the diagram is explained in one-liners here. So when the axon terminal and other cells, they are separated by a narrow space, that space is known as the synaptic cleft. So when an action potential reaches the axon terminal releases molecules of a chemical which is known as the neurotransmitters. What these neurotransmitters do? So these neurotransmitters, they travel across the synaptic cleft and bind to the receptors on the membrane of the other cell. So if the other cell is a neuron, it will start as an action potential in the other cell. So this was the process of the transmission in the synapse. So these are some of the references.